everyone. Thank you for joining me today. Also, for the people online, thank you. And especially thanks for the organizing committee for giving me the opportunity to present my research here today. I'm a postdoc at the Integrated Biology Department at UC Berkeley with Professor Britt Koskela, and I will be talking today about local adaptation. And for the sake of time, I will reduce myself to the phage side of stories. And as a study system, I'm going to the next slide. Yeah, okay, great. And as a study system, we look into pear trees, and more specifically, the ornamental pear trees that are all around here in Berkeley. So they're not regular pear trees, they're not commercial pear trees, but they're pear trees that are planted in the 60s and the 70s because of their nice flowers. But those pear trees here in Berkeley have a problem. They suffer from a disease called blossom blight, also fire blight, but in this case, blossom blight. And blossom blight is a disease that manifests itself, as you can see over here, by the leaves turn brown and black, the young fruits in this case turn completely black, and the bacteria themselves, they have a taste for flowers. That's also why the disease is called blossom blight. And the disease specifically is caused by Pseudomonas syringae, Pathovirus syringae. And as you can see over here, if you inoculate them in pear fruits, they kind of become quite nasty. So this disease is actually a really big problem in commercial pear production as well. And in our lab, we have isolated over time 2 different strains, PSS 16QS5, which was isolated in 2019, and we have our PSS 11.5 isolated in 2022. Now, additionally, with our PSS strain 16QS5, we also isolated a bacteriophage called 16Q. Coincidence? Not. And this phage over here, if we whole genome sequence the phage, we see that it's actually a novel phage. It has not been described before. And if we look into the genome comparison to the most related phages over here, we see that the phage shares less than 75% of sequence identity with its closest neighbor, which confirms that it's actually representing a new phage genus. If we then zoom in into the genome itself and compare it to the most related phages, PBH09 and KIL5, we see that the genome architecture is nicely conserved between the phages, and we see that these nice clusters of, for example, here the structural proteins appear, which will become later important in my talk. So we have here this nice system of a sympatric strain of PSS and a phage, and an allopatric strain that was isolated a few years later. So this gives us the perfect playground to ask ourselves questions on how does local adaptation influence phage effectivity in a biocontrol setting, and how does coevolution influence these principles of local adaptation. And this is what I will be talking about today from the phage perspective. So in order to figure out how this local adaptation is influencing our phage infectivity in planta, we designed a bioassay based on immature pear fruits. So what we do is we take unripe pear fruits from the tree, we surface sterilize them, and we poke literally 3 holes inside each pear, and then we inoculate our phage inside. We let it sit for a while so that the phage itself is absorbed by the fruit, and then 1 hour later, we inoculate them with either our sympatric strain, PSS 16QS5, or our allopatric strain, 11.5. We incubate our phages over time, or our pears over time, 5 days, and then we read out the amount of massacred tissue that is surrounded by our inoculation zones. So basically the necrosis. And how does this look like in real life? These are the pears that we have. These are the ones with the bacteria. These are our negative control with our buffer condition, and this is a combination with a phage. So if we then read this out in practice, we get this. So over here, we have our control condition, which is just the buffer condition. We have a little bit of browning of the tissue, which is due to oxidation, which is quite normal. Remember, if you cut open a pear or an apple, it becomes brown. This is basically the same principle. Then over here, we have our allopatric strain 11.5 and our sympatric strain 16QS5. If we add no phages into the system, we see that we have a nice infection going on for both 11.5 and 16QS5. And if we then add our phages, we see that we have a significant disease reduction for both the disease caused by 11.5 and 16q-5. But what was striking to see is that there is actually a really big difference between the disease reduction within our sympatric system compared to our allopatric system. So this made us think, is there an effect of local adaptation into these systems between the sympatric and the allopatric strain? So in order to figure that out, we set up a co-evolution experiment in vitro in this case. And what we did here is we co-incubated our phage together with either 16q-5 and 11.5. We incubate this for an extended period of time. And next we dilute this co-culture 1 in 10 into fresh medium. And this is our first passage. And we did this up to 5 passages. And we looked at our phage concentration in function of passage. This was important because if we see that there is an adaptive, or if our phages are adapting to the bacterium, we can hypothesize that the concentration of phage maintains rather stable over time. Whether if there is no adaptation, the phages get out diluted from the system because we're always diluting 1 in 10. So if the phages are not able to break the resistance of the bacteria, they get out diluted from the system. If we then look into our phage concentrations over here we have our sympatric combination, here we have our allopatric combination. We did this experiment for 4 lines in parallel. What we see is that in each of the 2 systems, we see that 1 line gets out diluted. So it is not always the case that our phages are, in this case, adapting to the resistance that is developed by the bacteria. So in this case, we have our line 4, and in this case, we have a line 3. But in general, in the other 3 lines, we see that there is a coexistence between the phages and the bacteria. Even though the bacteria develop resistance, the phages are able to break this resistance. So we collected our phages from this system because, of course, we were curious to see, do we see mutations accumulating into these phage genomes that give us a sign that there is indeed a local adaptation happening into this coevolution experiment in vitro. So we whole genome sequenced our phages from every line, and we were very happy to see that indeed we found mutations. I was thrilled to see these mutations. I've done a lot of coevolution experiments, and this was actually the first time that I really saw mutations happening in my phage genomes. So I was super happy to see it last week. And what we see is that the mutations that are occurring into our PSS16q5 are mainly located in this area over here. If we then look into the gene products that are encoded here, we see that they're mainly located into the tail fiber region of the phage, which is not that surprising if you think of it, because this is a vertical evolution system. So the mutations that the bacteria acquire are likely to happen on the receptor level. So the phages are adapting to our resistance. Now, what was interesting is that there is also a difference, and we saw a difference in the SNP analysis between our sympatric combination 16q5 and our allopatric combination 11.5. We saw that again, the mutations in this tail fiber region happen, but for 11.5, we see that there are additional mutations in the base plate occurring, and also in the GDSL-like lipase. Here also in DNA polymerase and so on. So there are differences from our phages to adapting to the different strains that we have. But of course, the next question that we asked ourselves is, what is its relevance of these mutations? Do we see that this local adaptation is also represented in the efficiency of our phages to limit the disease outbreak in the pairs, right? So what we did is we combined our phages from the evolution lines together, and we did again our infection assay. And let me walk you through this graph here. Again, we have our control with no bacteria inoculation. We have here our allopatric strain 11.5, and here are co-evolved phages. And we see that there is basically no difference in the disease reduction for either of these coevolution lines. But what we do see is that our 16q phage that is adapted now to our allopatric strain is actually less efficient in controlling the disease for the ancestral 16q5 phage to which it was originally co-isolated. So we see here that there is a big difference. So we still see disease reduction, but there is a big difference between between its ability to reduce disease from 16q5 versus to R11.5. And this suggests that local adaptation in these coevolution experiments is actually confirming what we saw before, that there is a difference, and that phages are indeed adapting to their host, and that this is of importance for a biocontrol setting. So this is it for me. I would like to thank, first of all, our funding agency, but especially my advisor, Professor Britt Skella, 2 grad students that were involved in initializing the project, Eli and Emily, our postbac researcher, Sarah, and especially my team of undergrads who are my helping hands and who are amazing, Saray, John, Sydney, and George, for all of their hard work in this project. And of course, I would like to thank you for your attention, and I'm very happy to answer any
questions. Yeah, that was fantastic, by the way, and really beautifully presented.
Thank you.
So all the phages and the syringe was isolated from the same tree?
They were isolated from 2 different trees. So we have our tree 16 and tree 11,
okay.
And they were from different years. So this really makes us convinced that we are talking about this allopatric system where they're not actually coexisting with each other in nature.
Correct. And then you see the seasonal changes, as Britt has already done some of the work earlier. Do you see the seasonal changes in these strains also? Like, do you find the same strain with the mutations? Does that match with your evolutionary experiments?
That is something that I have the sequencing data for, and,
okay.
and I'm doing the analysis right now, but that is true. So we're actually sampling our pear trees in function of time, and we're seeing into these effects of local adaptation over time. This is like a very small snapshot of the initial coevolution experiment in vitro, and now we want to see how these results correspond to what we actually observed in nature. Yeah.
Fantastic. Thank you.
Thank you.
Great. All right, we've got a couple. Let's see if I can make it back here. Maybe we'll pass the mic down if you all don't mind.
Really good talk. So when the mutation, the SNPs you're seeing in the tail fibers, do you know if they're synonymous, synonymous mutations?
Yeah, they're non-synonymous mutations. So there are amino acid translations into these mutations. Yes.
Oh, great, cool. Thank you.
Yeah, they're non-synonymous mutations.
Excellent. Is there? Yeah.
Is the phage
No, it's a
into the
lytic
bacterial gene?
phage. It's a
Okay.
lytic phage. Yeah, yeah. This is a lytic phage.
So what's the next thing that you're excited to do?
Oh, so many things. The next thing that I want to do, so I actually know that this phage is not only present in this specific time point from which I isolated it. With qPCR data, I was able to track the phage down in function of time. Now I actually want to see what the mutations are in these phage populations to see if this relates back to the coevolution experiment that we did, because of course the bacteria here in the system are super happy. They're growing in a rich medium. There is basically no selection occurring. But in a natural system, you have other bacteria that are competing with your organism. You also have the selection pressure of the plant itself that is actually influencing the ability of creating all of these mutations or mutants, because we can hypothesize that they're less competitive compared to the other native non-mutants and so on. A small snapshot. So I also did the bacterial side, and we see that the mutations are occurring in the LPS, so genes that are responsible for LPS production. So we know that LPS is one of the major virulence factors for Pseudomonas. So we can actually hypothesize that these guys are less virulent in this case, so that their competitiveness compared to other microbes is less.